Thank you very much. My talk was introduced already twice. Well, it's a pleasure to be here, of course, and I'm very grateful to the organizers, particularly to Gabashamlai, for making me coming to this exciting conference. I've learned a lot already. I don't know if you will learn a lot from my talk, because it's a completely different subject we study, such as mice, but I will try to explain you how we were doing this. Okay, all right. So, yes, the study of molecular mechanisms underlying late life depression. It will be not only late life depression, but I hope we will have time for additional data beyond this. I put this picture of a beautiful lake, Balaton, not only to embellish the beginning of my presentation, but also to share with you quite, I would say, slightly disturbing discovery I made with our beloved ChatGPT. Because when I asked how much of the deuterium can be found in Balaton Lake, I got a quite vague response. Tens of thousands of liters, but also I was told that, and I think you will agree with me, that the chemical impact at normal levels, biological impact is negligible. So I think for all of us in this audience, this statement sounds a little bit disturbing. So what we were discussing yesterday, the idea that actually the awareness about the importance of this topic has to be improved by far. We can see that ChatGPT only can confirm this, in my opinion. All right, so I think I put some slides, general slides about physical properties of deuterium-enriched and deuterium-depleted water, but I think it's pretty unnecessary in this audience, especially after having excellent lectures we have had yesterday on this, also on the cellular effects of altered concentration of deuterium, isotope effect, proton-coupled electron transfer reactions, mitochondrial respirations, and mitochondrial bioenergetics. I think it was all discussed by real experts yesterday, but all these have prompted us to perform our studies, which were, of course, also based on the evidence of importance of the consumption of water in humans. So where actually we consume not negligible amounts of deuterium daily. That's probably, ranging between 0.7 and 1.2 milliliters, so maybe 1 ml per day. That is not negligible. That was also mentioned. So once again, the geophysical processes resulting in the huge natural variation in contact of deuterium in water were also extensively discussed yesterday. We were referring to the continental effect, where the concentration of deuterium in drinking water would, as we know, decay in proportion to the distance from the ocean. And based on this, so there was a study done quite some time ago, and maybe you've seen this picture already, but let me remind you this one, because it was really a starting point for our mouse work. So that was a colleague of us, Gerpamitkin, who prompted quite a large investigation, a study done by the University of Utah at that time in the United States, where the concentration of deuterium in tap water was related to the incidence and prevalence of all possible medical conditions. And one of the interesting findings we have gotten from this was obviously high correlation between deuterium content and wrong pointer. Deuterium content, as you could see, in areas of the United States which are, well, kind of a shore part of it, and prevalence of major depression. So that was one of the findings, quite striking findings that our colleagues got. Another one was also absolutely striking one, was a finding of a significant correlation between prevalence of diabetes type 2 and deuterium content in tap water. So based on that, we were quite intrigued whether these correlations, this phenomena, could be reproduced in mouse models of respective disorders. Right. So for that reason, we were trying to use actually in our work mostly water that would be containing still natural levels of deuterium to mimic natural situation. So the water we were using was ranging between 90 ppm, that is lowest possible we found deuterium concentration in drinking water, and 180. We also have done work with extremely high levels of deuterium, such as 300, but, well, these studies were also very helpful, but quite unfinished for now. So we were using models of chronic stress to mimic depression, to induce depression, aging, that is also related to development of late life depression, major depression, and so-called Western diet. And I will explain these models and how we were treating our mice with all the waters we were using. So first of all, depression. In a generally accepted classification, depression is defined by presence of at least one core symptom that is typically accompanied by a number of subsidiary symptoms. And those core symptoms of depression are depressed mood and so-called anhedonia, decreased sensitivity to reward. So, of course, depressed mood is something we can't use in a mouse. We can't ask a mouse to fill out a questionnaire. So, but what we can do, we can still assess the sensitivity to reward. We also use additional measures, behavioral measures for depressive, well, not depressive-like behaviors, but accompanying depression features, such as reduced ability to cope with challenges, helplessness, reduced interest to novelty, cognitive impairment, sleep disturbances. To study late life depression, we investigated quite aged mice. So mouse normally lives maximum 24 months, right? And so the young age is considered to be like 3-5 months of age. So we were using quite old mice, 18 months old mice. Such studies are very difficult, challenging to perform in Europe for all kinds of reasons. It's very expensive. And just to have a cohort of mice of this age, of same gender, so this is already a challenge. Well, quite some time ago, we were studying these old mice, and we found that, in fact, they do display features of depression, such as anhedonia, as I mentioned to you. How do we measure anhedonia? So the test is kind of simple if you know how to do it. So when the mouse has a choice between sweetened water and normal water, a happy mouse, as you can imagine, could go for sweet water, of course. But actually, when the sensitivity to reward goes down, we have a decrease in sucrose preference in this test. That's quite a nice test, but, well, again, you need to know how to do it. Another thing we found in these mice was a decrease in exploring novelty. Okay, there were some other elements in this, but I will just go straight to molecular changes we found. Well, it was a little bit old, kind of outdated technology we were using. We done Illumina study, and just comparing these old mice versus young mice, we found that in the limbic system of animals and hippocampus in particular, so already the age produces quite significant changes in gene expression. And these changes concern such features as immunity, chromatin remodeling, regulation of mitochondrial processes, cancer. Okay. So more changes was found in the prefrontal cortex that mostly regulate emotionality, but also memory. And again, so well, we got quite a long list of genes. And again, so we mostly found genes that are involved in regulation of immunity, DNA repair, mitochondrial function, oxidative stress. So then we were quite keen, based on a lot of previous data, but also quite nice findings coming from Gabor's group, showing that and I think yesterday this data were shown by Roman also, showing that under conditions of manganese toxicity, nematodes could live longer if they are housed on the deuterium-depleted water. So that was quite a striking finding for us, and therefore, all right, so some genes accompanying these changes were identified. So we were curious whether the exposure of this quite old mice to deuterium-depleted water, and again, we were really using natural, let's say, concentrations, the lowest possible that could be found on Earth, 90 ppm, whether it can exert any effect on depressive-like behaviors. And yes, to our surprise, only a 2-week housing on this water already resulted in increased sucrose preference. That was quite an important finding for us. Another thing we were measuring was helplessness. And again, it's a kind of a simple test in a mouse. So it's where you place a mouse in inescapable situation of swimming in a water tank. It can display either struggling or a passive floating on the surface. And this floating behavior is taken as a measure of behavioral despair and helplessness. And this helplessness behavior, as you could see, was significantly lower in mice that were housed on the deuterium-depleted water. So the novelty exploration was also improved in old mice that received deuterium-depleted water in more than one task. This is actually a real object that we use for a mouse. It's not easy to find objects that would be interesting for a mouse to explore. by the way. Yes. Yeah, well, this is a very important finding, I think. We were also testing memory function in the mice. Well, as I mentioned, memory impairment is an important feature of depression, and especially, of course, it's also associated with aging. So we've done a classic test for hippocampus-dependent memory, contextual fear conditioning, where a mouse is placed in electrified box and well, with certain visual cues, and receives unpleasant electric stimulations. Next day, if it recalls the whole context and situation, it would freeze in response to this exposure to this box, as it does in the natural situation when, for example, it's confronted to a predator. So it's a reflex to freeze, to hide. So by this measuring the freezing behavior, we could then measure to which extent the mouse recalls the context. And while old animals, not surprisingly, remember very poorly the situation, in case when they are housed on normal water, old mice that received the Deuterium-Depleted water were much better. So they had improved memory in this test. And again, of course, we were interested in possible gene expression changes, molecular changes, potentially underlying these behavioral effects. So we've done, again, well, quite old-fashioned assay with Illumina, but still was informative, looking at the gene expression in the hippocampal formation and also prefrontal cortex. And yes, we found significant differences between the groups. So, but when we would just look at altered gene expression with more than 1.5-fold change, we would have only a few genes found. This is not surprising. So, because we had naive mice, which were not manipulated, so the effect was supposed to be minimal. We had water of with natural range of concentrations of deuterium, and yet we found some differences. But also we were quite happy that we didn't have so many genes, because for us it was showing that mice were not infected, as often can happen in facilities, so we could actually rely on the result. Yes, and we found changes in the prefrontal cortex. So those were actually different genes from those that we revealed while comparing young versus old mice. But still, those were genes related to DNA repair, oxidative stress, immunity, and also this time circadian regulation, such as PER2 gene. So we found more genes altered in the hippocampus, and there were also similar functional properties. Just to give you a couple of examples, the most studied genes are here. Early growth response 1. So this gene is involved in pathology related to aging and Alzheimer's pathology, for example. So the other one is CRIMP1, also was found to be linked to aging and an NMDA receptor-mediated loss of synapses. Yeah. Okay, well, similar direction of changes with other genes, and we proved the gene expression changes doing additional PCR. So from that, we could conclude that yes, actually, the Deuterium-Depleted water of 90 ppm can counteract manifestations of late life depression behaviorally, but also it alters gene expression. Yes, and this gene expression mostly related to immune responses, oxidative stress, cellular plasticity, aging, circadian regulation, DNA repair. We were quite pleased actually to see that the scope of the gene expression changes was keeping with recent findings, publication coming from Gaber's group, where different genes, of course, were found in a completely different setting. That was work done on the lung adenocarcinoma cells, but still, so we found like a similar direction of the changes. All right, okay. Another type of model we were using was a chronic stress model. So where mice, in this case, actually young mice, were exposed to a series of stressors. For example, rat exposure, and rat is a predator for a mouse, so it's a threatening element, and other unpleasant procedures, short tail suspension, restraint stress, and this all little stressors we found can result again in induction of anhedonia. As I explained you earlier, it could be determined in the sucrose preference test. And I shared with some of you our findings that in this case, not all mice develop this syndrome, but some of them remain resilient. And that's an interesting phenomenon per se to also compare what makes mice resilient here. So that's what we were doing, giving our animals, again, deuterium-depleted water of 90 ppm, while exposing them to this chronic stress and testing them in the sucrose preference test. In this case, we had a pharmacological standard, a classic antidepressant, citalopram, SSRI antidepressant, and we actually found the effects of rescue of sucrose preference that would be comparable to the effects of citalopram. That was quite nice. In the swim test, again, we found a rescue effect on floating behavior. All right. And on the gene expression level, we didn't do much, but we found, very importantly, that expression of serotonin transporter, one of the key molecules involved in the mechanisms of depression, was actually also rescued by the treatment either of antidepressant SSRI or deuterium-depleted water. This time we had a chance to study EEG sleep in these animals in a specialized lab in Lyon, because, well, we're doing this because it was quite important to study REM sleep, since this is, well, depression is the only disorder which could be diagnosed by changes in sleep, in EEG sleep. And this change is an increase of REM sleep. So we were interested whether REM sleep would be altered. And yes, REM sleep was normalized, was decreased. But also we found effect on slow wave sleep and wakefulness that were increased in these animals. But that's maybe a separate story per se. Yeah, so that was to us quite convincing that in this paradigm, deuterium-depleted water induces antidepressant effect. In addition, we've done a short experiment using stress of rat exposure, where we assessed hippocampal cell proliferation, because in depression, so we all know that the hippocampus getting shrinked, and you have a suppression and getting atrophic. So we found that using two markers that in fact neurogenesis or cell proliferation in ventral part of the hippocampus is preserved in stressed animals that received deuterium-depleted water. So that was our conclusion. I will have still a few minutes to share with you our latest results, and this will be the last part of my talk about actual metabolic syndrome or diabetes type 2, because this is if it's not a depression, but it's the comorbidity between diabetes type 2 and depression is high, and it was a very exciting finding, and I'm showing this again, of a correlation between the deuterium level in drinking water and prevalence of diabetes type 2 in United States of America. So therefore, we were also very keen to perform the study. And again, so we were quite, partly triggered by the results that were presented yesterday by Gaber, who used a model of diabetes in rats, and then was using different concentrations of deuterium in water. So we were using so-called model of Western diet. Western diet is a nutritional style characterized by preference to highly caloric food, which is enriched with saturated fats and sugar and cholesterol. This is something opposite to Mediterranean style of eating. And this can be also modeled in mice. So in mice, we found it causes absolutely determinous effects. So the model was in fact developed in Maastricht University by our colleagues from Department of Physiology, but they never looked at the brain. So for some reasons. In our group, we decided to study what happens with brain. And we were quite shocked because it's a lot of things is happening. So yes, Western diet, housing of animals for 3 weeks on so-called Western diet, and we buy it as a standardized diet in research diets from the US, induces glucose, so impairment of glucose tolerance, but also we found that in the brain, we see upregulation of inflammatory molecules, such as toll-like receptor 4, for example. And the downregulation we see in the brain and in the liver of mitochondrial markers. Steatosis in the liver, and also inflammation, and a lot of other things. So this is not Western diet, this is foie gras. Of course, we did not feed our mice with foie gras, but yeah, that was Western diet standardized and bought from the US. All right. Okay, yes. So we were trying different concentrations of deuterium. So this was a pilot experiment with 300. I will not show it this time and also don't have much time. So, but basically, we were using old mice, females. So it's a kind of trying to recapitulate a cohort that is at high risk of metabolic syndrome because of age, and because the mice were already at menopause, they were 12 months old, and we wanted to know whether the natural variations, extreme variations in deuterium in drinking water would actually exert any effect on these animals. What we found that actually, yes, when we use deuterium-depleted water, we could actually, Western diet would increase the glucose level in the glucose tolerance test, meaning impairment of glucose tolerance. But this did not happen in mice that were housed on deuterium-depleted water. Yeah. I have to say that it's very difficult to beat the effects of Western diet, especially in aged mice, especially in females. But deuterium-depleted water can do that. Yeah, so same experiment, just different time points. What was nice to see and I mentioned this earlier, this effect just on old mice, that actually in the object recognition test, where a mouse is supposed to recognize a new object in this 2-day test, we found improvement of the object recognition behavior in our old mice receiving deuterium-depleted water. I think this is a very nice result. So, yeah, and strikingly, the liver steatosis was not improved at all. So it's really, so this water counteracts the very kind of strong effects coming from peripheral inflammation. This is associated with liver steatosis. Yes. And then we were trying to do just the opposite. Again, used mice of that age, around 12 months old, housed them on Western diet, but this time we used another extreme regarding deuterium. So the deuterium level was 180 ppm. And you can maybe notice that the peak of in the glucose concentration in blood in glucose tolerance test would be observed a bit earlier, and without actually significant overall effect on the glucose tolerance, but so that was difficult to say, but okay, so there were some signs of changes in this test, but not significant. So the shift towards the higher level of deuterium did not strikingly alter the glucose tolerance in this situation. So again, we looked at the steatosis, and it was not altered. But what was clear that the exploration, novelty exploration, was suppressed, clearly suppressed by by the use of deuterium-enriched water in these mice. Also, what was important is that hippocampus-dependent memory, in this case it was step-down avoidance test, was suppressed by the use of deuterium-enriched water in these animals. So where normal mice would acquire the test, Western diet would suppress the learning, and this effect is well known because, well, as I showed you, it induces massive inflammation in the brain. And in the step-down test, so mouse is placed on the platform. Once it gets down, it gets unpleasant electric shock. And then if you place it back next day, and it goes down, it means it does not remember the context. So that was exactly a case with the short latencies in mice that were housed on Western diet or received a Western diet and deuterium-enriched water. So yes, deuterium-enriched water seems to be does not significantly affect glucose tolerance, but it suppresses memory. It's almost the end. So this is a test of novelty exploration again. And again, we saw suppression of novelty exploration. This test, well, and this is my last slide, again is about hippocampus-dependent performance. I don't have a picture here, but mice have a very funny, behavioral feature. If there is a cylinder with small object, they tend to displace the object. Nobody knows why they're doing this. This test was developed in Oxford a long time ago. But what is well known is that if you destroy the hippocampus, they stop doing this. And, or they do it very slowly. So that was the case with mice that received deuterium-enriched water. So they were not able to perform this task. So another confirmation that actually seems to be plasticity suffers on the first place rather than metabolism with deuterium-enriched water in aged animals. That was, to us, it was a surprise, really. I guess that's all. I think all the conclusions were already outspoken, and I would like to thank you for your attention. And I have to mention a lot of people who were helping with this work. Some of them are in this audience. This work mostly was done in Lisbon University, where I was affiliated partly as a joint position for quite some time, but a lot of other places, Wisconsin University, Oxford University, Claude Bernard University in Lyon were involved in this. Thank you. That's all.
Thank you.